Right, cool. Okay, so today we're chatting with Thomas, who does really, really cool work on astrocytes. And Thomas, you probably won't know this, but the way we first discovered you and your work was from this beautiful perspective piece that you published with another author, Murphy Royal, I think in 2023. And it's this beautiful perspective about trying to kind of find a comprehensive, I guess, framework for thinking about astrocytes. And I came upon that, in 2023, and I actually shared it with the group here. And most people, including Mac, we read it and we loved it. And we even I remember Mac even making a note saying, oh, we should, put this in a file of good examples for students for, really nice perspective pieces, because it just had a great combination of cool ideas and was written, in a beautiful way. And then fast forward a couple of years later, I've been lately in a hell of my own making, trying to write a comprehensive review about noradrenaline and noradrenaline in learning. And I realized that one of the most exciting things about kind of what noradrenaline, the new evidence we're finding out at the moment, is this possibility that kind of astrocytes and noradrenaline are working in concert with each other to shape even things like really, sort of complex behaviors like learning and that sort of thing. And that's what led me to read back your perspective piece. And I discovered this beautiful recent preprint that I think you might talk a bit about today. And then I emailed you to ask if it was okay if I adapt one of your figures. And then you came back with this, beautiful, enthusiastic chat about all things astrocyte and noradrenaline. And that was when I knew you would be a great person to talk to. So without further ado, if you want to kind of get started, I don't know if you have a few slides or, you want to kind of start a pitch of things. Otherwise, we can start prodding you, whatever you think.
Sounds good. Thanks for the generous introduction. That's a very polite way of you to say that I basically got carried away in my emails and
exactly
and wrote very long emails when you were just asking a yes or no question. Let me start maybe by asking, can I just have a quick show of hand? I think I can see everyone who really doesn't quite know what an astrocyte is and what they look like.
I'm keen to learn more about what they look like and what they are.
Yeah, I'm not going to go into too many. I don't want to go into too many details about what they do and how they do it. But I think it's always useful to start with what they look like, because I think it tells you a lot about what they might actually do. It's the old concept of structure-function relationship. So let me try and see if I can share my screen. Here we go. Where do I start? This is very unprepared, as you can tell. I'm sorry. Up. Thank you. All right, can you see my screen?
Yep.
Yep.
All right. So this is going to be very off the cuff, but this is obviously not an astrocyte. I think you can all recognize a neuron. And I don't know how well you can see on your screens, but I hope you can appreciate, obviously the soma and the dendrites and the very fine dendritic spines and the neck of these spines. And I think for all of us doing neuroscience, this is kind of the reference we have. This is the most refined and small and compartmentalized as you can get, right? A dendritic spine and the neck of a spine. And just to give you a sense of what we're talking about here when we talk about astrocytes, this is an astrocyte just nearby that same neuron that was obtained through serial EM and 3D reconstruction. And I'm going to zoom in a little bit because I don't think you can fully appreciate. Yeah, this is pretty much as zoomed in as I can get without it getting blurry. But I think you can really realize, I hope you can anyway, on this image, how intricate the morphology of an astrocyte is. It's really made of a bunch of ultrafine processes, by lack of a better word, that seem to mingle with all elements in the nervous system pretty much. They seem to be just this massive, I don't know, some people call it a bush-like structure made of an infinite meshwork of very fine processes. And I think that the best way perhaps to really get a good sense of this is fairly small saturated reconstructions of brain volume. So if you take just a tiny chunk of that astrocyte and you reconstruct with EM all of the elements in that volume, so that would be something that's about 5 microns by, I think, 15 microns, you can somewhat extract just the contribution of the astrocyte to that volume. And you realize quickly that you have to deal with a cell excuse me, that really seems to infiltrate all the nooks and crannies of the extracellular space that exists between all other cells. So it doesn't even quite look like a cell, frankly. It looks almost like if someone had injected resin into the extracellular space in the brain, waited for it to harden, and then melted all the cells, and you're left with this pretty much. Right? Obviously, this is a very bad analogy for someone who's trying to advocate that glia is not just glue between the other cells of the nervous system, but that gives you a sense of what these cells really look like. You don't have the typical neurite or axon or dendrite. It's just a very complex, finely regulated meshwork of ultra small processes.
You know, Tom,
And so,
your head what the
go
Latin
ahead.
for resin is?
What was that?
Do you know off the top of your head what the Latin word for resin is?
I don't actually.
We should look it up because we can just rename it that.
Yeah, that would probably not be any better than glia, but anyway. So the reason I want to mention this is because if you have this view of astrocytes as interpenetrating all of the crevices of the extracellular space, it tells you quite a bit about what they might do and how they might do so, because it really advocates that these cells are probably surveilling or listening to everything that's happening in the extracellular space, which is just a medium for signals to be channeled between cells. But it also tells you that maybe they control that space in itself. And I think that was kind of the starting point for the contextual guidance idea that we developed. And I think it also makes a lot of sense in the context of neuromodulation. And I think if you also study norepinephrine, you'll probably know why. So let me then skip maybe straight to the review we wrote. Sorry, let me stop sharing my screen maybe. I'm just going to go maybe through some of the figures of the review, and I think then we can just go straight to questions or discussions, unless you want me to talk about some of the unpublished stuff we have.
This is great. Let's keep going on the complexity of the brain as it is. We've recently been playing a bit with the microns data, in particular, Jason.
Oh yeah.
And regularly in lab meeting, we just get Jason to pull up exactly the same kinds of pictures that you have been showing, and just marvel at the complexity and think, man, I've got this little toy idea in my head of a neuron as like a little triangular cell body with a little line coming off of it as a dendrite, with some branches, and an axon coming off as another line. And then you get down into the level of the actual micro scale, and it's all bets are off. Your understanding of it is just completely lost. So this is really, really helpful.
Yeah, I've been playing with the microns data as well. It's a bit frustrating from a glial perspective how little they did with glial cells, but I think there is enough data there to mine for a few years for sure. So the idea of the review we wrote, well, the perspective, I guess, was that when it comes to brain states, the way brain states are really fundamentally regulated is through, at least in our view, two main ways. You have circulating signals, and you have essentially neuromodulation stricto sensus. So those circulating signals are going to be things like, hormones, stress hormones, or metabolized glucose, oxygen, CO2, things like this. And what's interesting is the first cell that you would have to encounter if you were one of these molecules crossing the blood-brain barrier is astrocytes, right? Not neurons. Astrocytes can sense oxygen and CO2. They sense hypoxia, hypercapnia, and they do so in particular in areas of the brain that regulates breathing. And then they also sense things like glucose and insulin. They have insulin receptors, and they particularly do so in brain regions that regulate feeding. And so you can rapidly kind of generalize this idea of astrocytes sensing particular signals that carry context and brain states. And then that brings the question of, do they participate in the regulation of these brain states, or maybe in the way that these brain states impact neuronal activity? And then in terms of neuromodulation, you have the same idea, right? We know that brain states go along with changes in norepinephrine, acetylcholine, oxytocin, or a bunch of neuromodulators. And it's interesting how astrocytes are probably better equipped or better positioned or better shaped to sense these molecules and neurons. Because as you all know, neuromodulators are released. They're not released like in a synaptic way, like we always see it in schematics. Like you don't have a terminal impinging on like a neuronal element. It's actually more like a very sparse strings, right? Axons with varicosities here and there that really is neuromodulators in a fairly an homogeneous and untargeted way in the brain. And if you're a cell like astrocytes with ears and hands pretty much everywhere, it's much easier to sense variations in neuromodulation tone than it is if you're a neuron. And so we started having this idea that maybe astrocytes are sensing all of these signals. And then once you have that in mind, the rest just flows by itself, because we know astrocytes have many, many ways to regulate neurons and neuronal networks. And so that gives you a schematic right away where astrocytes might be a gate or an entry point for neuromodulation or hormones and how they impact neural circuits. And I think that's pretty cool too with this view is that, well, I guess in a way neurons are relatively simple in the sense that they're unimodal. Like if you're a glutamate neuron, well, I know what's going to happen if I activate you. You're going to release glutamate, and glutamate is excitatory. Astrocytes are multiplexes. That means that we know many things they can release, but they can potentially release all of them. Some of them are excitatory, some of them are inhibitory, some of them are just modulatory. And so that gives you a very complex way now to think of the effect of neuromodulators on neurons and circuits. And because astrocytes also contact different parts of neurons in different ways, like they can contact synapses, they can contact the soma, it gives you a sort of a manifold way by which neuromodulators and hormones might be able to impact different parts of the neurons with different functional outcomes, right? So we think this can add a lot in terms of richness of the effects and the heterogeneity of the effect of neuromodulators and hormones. And go to maybe the next figure quickly. Yeah, I guess one way this can be interesting, at least from the point of view of synapses, is that if you think of all of these things coming together, it gives astrocytes a way to maybe reshuffle functional connectivity, which is of course central to the idea of neuromodulation and how neuromodulators impact brain function. And so we started playing with this idea of like a having a fake little circuit that astrocytes could modulate when a neuromodulator comes about. And of course, it's very simplistic, but you can imagine how this could actually be true, right? You have a circuit that is shaped to do a specific thing where the flow of information goes from the input to the output here. And then once the astrocyte starts doing things to synapses or neuronal cell body and activity, you reshape the flow of information in a completely different way. So such that your circuit now is not doing this thing, but something completely different that is better fitted to whatever environment or behavioral condition the animal is in. And so that's what we call adaptation of the circuit, so adaptive configuration here. And the thing that's interesting, I think, with this view is that it places astrocytes in the driver's seat of how neural circuit may be reconfigured by neuromodulators, as opposed to just a sort of listener of what's happening at synapses. And so I think the final view here, the way to synthesize this is with this very simple figure that I think is really just a take on the set of what we describe here in this paper, which is that we go from a situation where astrocytes are just involved in a sort of weird feedback loop with synapses and neural networks in general that was fairly unsatisfying because you can essentially remove it and nothing changes. Things can still happen the way they did. But now what we propose is that astrocytes act as a gate for modulatory inputs or contextual inputs or brain states maybe to affect neural circuits. And now the main consequence of this is that if you take astrocytes offline, if you take them off of this schematic, things are going to stop not working in a fairly predictable way. You will predict that some modulatory inputs, for example, some neuromodulators, might no longer be able to modulate neural networks the way they normally do, simply if you take astrocytes offline. This seems like a fairly satisfying schematic for many reasons. First, we have a lot of evidence that some of these things happen. We know astrocytes can modulate, for example, synapses, and there are many examples of how they do this. The time scales also now seems a lot more satisfying than what it used to be before. We know astrocytes are somewhat slow. We know they act on the time scales of maybe hundreds of milliseconds, but more likely seconds, maybe minutes, which seems more fitted to the way that brain states occur and the time scales of neuromodulation as well. And we also know, we've known that for quite a while now, that astrocytes are sensitive to neuromodulators and hormones. So we thought this was a fairly, straightforward way of assembling these different pieces that also allows to produce maybe some hypotheses. And one of the hypotheses was that, well, the effect of some neuromodulators on neuronal circuits might require astrocytes or be less effective if astrocytes are not there. So that's one of the things that we try to test lately, and that's one of the preprint that we have on bioRxiv right now. I don't know if you want me to talk about that, by the way, but I'm happy to if you like.
I think it would be great to hear a little bit about the preprint. I have a question kind of just sparked off what you were, kind of talking about there, the structure and how astrocytes kind of fill up the space and contact a lot of things. It seems great for sort of sensing context, right? They can sort of, sense all these different signals from a wide area. But it seems really a really bad design for sort of more specific synaptic or neuronal modification, right? So and given that, we know astrocytes, they can have these more like wide-ranging effects, but they can also affect quite specifically synaptic plasticity and, learning and things like that. So yeah, astrocytes themselves, do they have sort of compartmentalized processes that can have these more discrete effects on synapses?
Yeah, this is a great question. I wish there was a simple answer. So let me just share maybe one more slide so we can talk about this with somewhat of a visual substrate here. We do know the contact synapses. We've known that for quite a while, actually. This is the first place that we all started looking at astrocyte neuron interactions. It's at synapses. So you have to imagine that some of these sort of like astrocyte processes extend and somewhat enwrap the synapse. How many synapses are contacted is still something that's kind of controversial, but I think the last highest resolution EM reconstruction that's been done suggests that as high as 96% of synapses are contacted by an astrocyte process. So you could imagine that virtually all synapses are, and therefore the synapse is not a pre and post synaptic element sort of situation, but rather a tripartite synapse schematic, really. But the question you're asking really is how compartmentalized is this sort of astrocyte arm around the synapse? Is it operating on its own? Is it getting input from the rest of the astrocyte, just like synaptic terminal root from the axon? I don't know. I don't think anyone knows. The coolest findings that have come about on this topic over the past few years is a paper from Keith Murray at McGill, where he used FibEM to reconstruct astrocytes with the highest resolution we've ever had. And then they did a lot of machine learning training to try and figure out if there was any sort of microcircuit level sort of architecture in astrocytes. Do they contact synapses with this sort of circuit motif logic? And it seems like they do. So they're not contacting synapses in a sort of random or homogeneous way. There seems to be an underlying circuitry in what synapses are contacted and how with respect to the rest of the morphology of astrocytes. But I agree with you. The question is, how much of what's happening within an astrocyte is compartmentalized versus under the control of the cell as an entity? It's not clear at all. What's interesting, though, is this idea of the tripartite synapse, so the idea that you have an astrocyte contacting both the presynaptic terminal and the postsynaptic spine. It's something we've known both from the standpoint of EM and from the point of view of function since the 1990s, right? And so this is also where this idea that astrocytes listen to synapses and then talk back started, even though I think it's very unsatisfying. But the thing that nobody really ever talked about is if the dialogue between neurons and synapses only happened at that scale of the synapse. So in other words, if the synapse talks to the astrocyte and the astrocyte talks back to the synapse, and you don't need any input from the rest of the cell, then from an evolutionary point of view, this little fragment of astrocyte would have broken up from the rest of the cell to become just a sort of perisynaptic cell, like we have in the periphery. You have perisynaptic Schwann cells that just do that. They take care of the synapse, regardless of what's happening around. And evolutionarily speaking, that's not what we see at all. We see astrocytes actually becoming more and more complex and contacting more and more things. In a human astrocyte is about 30 times the volume of a mouse astrocyte, even though the neurons themselves haven't really changed that much in terms of complexity. So short answer is, I don't have an answer to your question, which is a very good question, but I would say there is probably a cellular level, so a sort of integration of what has to happen in all of these different computational loci.
Can I ask a follow-up there, Thomas? I think it's really fascinating diving down into the microscale to kind of think about what the functional importance of a circuit could be as it is. And so I wanted to kind of push a little further there. So do you mind if I share my screen really quickly? Um, can I just do that, or how does that work? There we go. Hold on.
I reckon you should be able to just do it.
So there was this really lovely paper from Jeff Lichtman's group in Science, where they looked in a human cerebral cortex and then reconstructed at a nanoscale. I think they were probably competing a little bit with the Allen and Stanford to see if they could come up, and Baylor to see if they could come up with their own microns. But this is just this beautiful summary slide here. So here's this little tiny petavoxel, so a bit smaller than the one in the mouse. But then they've reconstructed the same sliver with the different types of cells and fibers and things. So over here you've got the pyramidal neurons, and then over here you've got blood vessels and you've got myelin. But little f here is the astrocytes. And this really struck me when I saw this image. I'd never really considered the notion that the astrocytes are almost sort of tessellating that space, a little bit like if I was flying over the top of a forest and looking at the canopy of the trees.
Yeah.
The astrocyte area, they sort of almost cover their own areas. But then I immediately then start to think just how much differentiation really is there. Is it more important that an astrocyte makes sure that anywhere there's space, I better be there so that if there's a blood vessel nearby, I can connect whatever neurons are nearby to that blood vessel, and therefore make sure that there's really no hypoxia, or that I can balance aerobic and anaerobic processes so that that neuron never really becomes in danger of developing pathological oxygen-free radicals and damage and such, and less important that I'm getting this particular neuron at this particular time. So in other words, the mesoscopic view makes me think of a collective behavior rather than a precise one-to-one behavior. And so I wanted to push back a little bit on this notion of astrocytes. I think you're absolutely right that they do play a state-dependent role in the process, right? They're selective to neuromodulatory ligands as well as neuropeptides. This is going to be extremely important. But whether or not they're actually involved in specifically modulating this neuron and not that neuron, and whether they have that pressure, I wanted to kind of get your vibe on whether or not something like this organization pushes back on that notion.
Yeah, so that's a very good question too. I guess it gets to the question of, is it determined what neurons are going to be contacted by what astrocyte and what synapses is going to be contacted by what astrocyte? It's probably not, right? I mean, the way astrocytes develop is that at the beginning they don't cover all of the brain. They slowly grow over the course of 3 weeks in the mouse brain until they start touching each other and then they stop growing. We don't actually know what the mechanisms are. It might just be some sort of chemotaxis and then repellent signals, but they stop growing when they touch each other, and that's how they end up tiling the entire brain. So you're totally right that when you look at them, it's like you're looking at the top of a canopy, in the sense that they really have non-overlapping domains. So if you are within the domain of an astrocyte, you won't have processes from the neighboring astrocyte. That area of the parenchyma is under the control, well, from my point of view anyway, of that one astrocyte and no other, right? Yeah. And so that actually might be a way that you can start implementing input-output rules, by the way, if you think of astrocytes as actively regulating neural circuits. The fact that you have a certain number of synaptic elements or neural elements, generally speaking, that are under the curfew of just one astrocyte means that you can have precise input-output rules, as in like, I sense norepinephrine, I'm going to do this on synapses. I sense acetylcholine, I'm going to do that on the cell body, things like this. But the fundamental question of, was this neuronal cell body meant to be contacted by that one astrocyte? It's a very good question. I don't think anyone has an answer to that at this stage. Honestly, I doubt it would be specified. I think it just happens that way, and then the astrocyte learns to deal with whatever synapses it was handed and whatever cell bodies it was handed, in a way.
So one clue that might, I think, point towards a different set of answers might be just how diverse are the molecular signatures of individual astrocytes in particular areas of the, let's pretend we're just in the cortex for now. We'll keep our minds there. So imagine that there's lots of noradrenaline receptors down on the left-hand side and then lots of acetylcholine receptors on the right, or something like that. You could imagine then that whether or not neuromodulatory inputs came into that circuit could then bias the system towards processing in the deep layers versus the superficial layers or something like that. Or, it could also be, you could also imagine a different organization, which is there's individual expression of these receptor types, but it's not spatially organized. It's kind of heterogeneous. It's very unlikely that two cells will have exactly the same genetic signature. That's something that biology really hates, right? Is identical things. It likes to have a little bit of variance all the way along. So you can imagine little bits of differences, but those differences may not be spatially prescribed such that if a neurochemical came in, you can now, let's say, hit noradrenaline and it'll activate a subset of the astrocytes, which will then hit a subset of the circuit, but not a precise spatial subset, and vice versa, acetylcholine. So it could be more about just generally letting the network know that something's changed in the state rather than actually prescribing it. And I think that would be a really fun thing to see empirical data confirm or disconfirm, I think.
Yeah, that's very true. And you can add another layer to this, which is something that's still puzzling a lot of people in the field, which is the notion of astrocyte networks. So where these astrocytes kind of touch each other at the border of their territories, how we call it, they don't just touch each other, they actually communicate through gap junctions. Well, communicate maybe is not the right term. We know they have gap junctions, and so technically speaking, some things can leak or pass from one astrocyte to the next through these gap junctions. The way we saw it very early on in the field is that if you patch an astrocyte, and in your patch pipette you put something like an Alexa, you'll see the Alexa diffuse in your astrocyte, and if you wait long enough, they'll diffuse to the neighboring astrocytes. And you can have an entire network like this of connected astrocytes. And the boundaries and the size of this network and the shape of these networks depend on what brain region you're looking at. So for example, if you patch inside of a barrel of the barrel cortex, only astrocytes within the barrel are connected. If you patch outside, they're connected to nobody, to no one, to no other astrocyte. In the hippocampus, you can have the entire CA1 light up like this by just patching one astrocyte. So there is this notion of network that maybe gives astrocyte the ability to exchange some signaling molecules, or maybe things like ions or glucose, lactate. It's completely hypothetical, but you could imagine that you now have a vast number of cells connected to oversee a circuit that's dedicated to a particular function. And maybe they're meant to respond in a particular way, like you said, to norepinephrine or acetylcholine because of the subset of receptors they express, and another network nearby would have different rules or different functions. But all of this is completely hypothetical because nobody knows what the networks are doing or what they're for. Even the idea that something actually passes through these gap junctions is still completely hypothetical.
Yeah. So in that way, that's really fascinating to know. So in that way, the astrocytes in a little bit more like interneuron populations, like GABAergic interneuron populations, than they are like pyramidal cells, the excitatory pyramidal cells. Because interneuron networks, like fast spiking interneurons, we'll talk about them a bit in the lab, like parvalbumin fast spiking neurons. They spike really, really frequently, and they're often thought to be involved in what we call feed-forward inhibition. So a glutamatergic input will come in, hit the fast spiking interneuron, and advance its phase, and then also hit a pyramidal neuron. And so what it does is it basically excites the pyramidal neuron, but then the fast spiking neuron then shuts the pyramidal neuron up. So it has a really punctate response, big EPSP, that happens in a short period of time. It turns out that those neurons are connected in the similar kinds of gap junction networks to one another in the same class, so the same genetic class. So they'll hit other fast spiking neurons and pass a little bit of their internal potential to the other neurons. So you can get this kind of waves that pop around the network when you play with models of these things in really, really fascinating ways that, again, I think we're kind of rapidly kind of hitting the limit of our empirical understanding of these circuits, because it's really, really hard to do high-level recordings of these things in awake animals in really interesting contexts that also have the specificity of the circuit that you're interested in. So we really don't know exactly how these things play out, but it's really fascinating to know that the glia, which work maybe a little slower and have different kinds of signaling molecules, have this similar feature to the interneuron networks of the brain, which we know is super important for feed-forward inhibition and other measures. So that's a really, really fascinating homology that I wasn't aware of.
Yeah, the one big difference though is that we don't have, from genetic point of view, subclasses of astrocytes. So in that sense, the network of astrocytes is not so much designed around a cellular identity, but rather, actually it's complete suppositions here, rather designed around what function does that network have in terms of controlling downstream neural circuits, right? So you can imagine that the two astrocytes, one that's maybe within the network and the one that is not, just right next to it, might have the exact same transcriptomic landscape. You can't say this is a type A versus this is a type B astrocyte. So we do have heterogeneity across the brain and sometimes across layers, but it's extremely subtle, and we see nothing akin to having a glutamate type neuron, an SST interneuron, a VIP. We don't have these sort of very clear-cut classes when it comes to astrocytes. So that makes things a lot complicated, first of all. It also puts limit to the sort of analogies like the one you just made, even though in terms of function, I agree that this could be quite similar on different time scales, though. Yeah.
On the idea of this sort of propagation through a network, I really like I think it's a paper from last year from Kira Pozkaz's lab, Cahill et al.
Cancer?
Yeah, cancer. And this idea that, a really sort of tiny injection of stimulation to one astrocyte causes this really broad, wide-ranging kind of spatiotemporal network activity of astrocytes. And I guess, yeah, if people are sort of, empirically looking at that, is it kind of an all or nothing thing? I mean, if you kind of, trigger an astrocyte, does it sort of automatically permeate over this network, or do you actually see this more kind of restricted, you only kind of, get activity in a couple?
Oh my goodness, this is such a good question. So basically, what is the input-output rule, or the dose response curve of astrocytes? We don't know. Nobody has really worked out like the activation function of astrocytes yet, and I think it depends on the output, and it depends on how you deliver the output. For some things like norepinephrine, it's also tricky in the sense that if you try to do it in a physiological way by activating, let's say, the leukoserous fibers, you get this wave of norepinephrine release. So you sequentially activate your network of astrocytes, but you don't know if this is because one of them got activated and activated everyone else, or if you're just following the sequential release of norepinephrine in the network. It's not something anyone has figured out yet. I don't know yet of anyone who's done, let's say, uncaging of norepinephrine locally to try and see, okay, if I essentially puff or uncage norepinephrine there, do I have the entire network that's responding, or am I just going to get like a local hotspot of norepinephrine response from that one astrocyte? Nobody has done that. It's actually something we'd like to do in the lab. And then norepinephrine is just a canonical example because you get such a massive response from all of the astrocytes. But if you do the exact same experiment with acetylcholine, you'll have something completely different, with just very focal activations of some processes of the astrocytes. And it gets back to the question you were asking initially. Do we have processing very locally in astrocyte processes, or do we have processing at the scale of the entire cell that then flows back into the processes to have very local outputs? It's still something we don't know. But the idea is there that you could potentially activate one astrocyte in the network and engage the entire network, even though nobody has actually proven that.
Yeah, cool. I might use this as a nice jumping off point to push you a little bit more on noradrenaline. And I guess just coming from this really nice preprint that you had in it, I think it's being published soon, with a complementary paper. And if I remember, there's, I think maybe in the abstract or the somewhere in the introduction, there's this sort of, tantalizing phrase about, upending 80 years of neuroscience knowledge and, really sort of think that, really, and I don't think that's an overclaim. I think it's a beautiful claim there. I think, encourage us to sort of think about neuromodulatory systems in a different way. And I guess, I wondered, what your sort of thoughts on that are, how these findings are pushing us to really think about these neuromodulatory systems differently.
Yeah, that's a very good question too. So I think what's important to keep in mind, and I'm guessing that the field of astrocyte people will not keep that in mind enough, is we focused on a very specific question, which was, what is the role of astrocytes in the effect of norepinephrine on synapses? And as you guys know, since you work on norepinephrine, it has effects on many different levels of physiology and networks, right? So we didn't look at the effect on, let's say, synaptic plasticity. First of all, we didn't look at the effect on excitability, on AHP, things like this. We completely ignored that. We just focused on one aspect, which is the effect that norepinephrine has been known to change the strength of synapses in a non-Hebbian fashion. So basically modulate either release probability or the amount of receptors postsynaptically, but something fairly rapid. And the question was, what was the role of astrocytes in that? And the question essentially came from predictions that seem to arise from the contextual guidance theory, which was, well, if astrocytes are this sort of link between modulatory inputs and synapses, then you would assume that some neuromodulators like acetylcholine or norepinephrine would impact synapses at least partially by signaling through astrocytes. So we were really excited to test that, and we had a bit of a fantasy of like, oh my goodness, can you imagine if it's all of the effect? It was kind of a running joke, like, what about if all of the effect of norepinephrine on synapses is astrocyte dependent? But it turns out this is exactly what we found. So we found that even in the most extreme case where you would take a brain slice or a chunk of brain and you bath apply norepinephrine at high concentration and you leave it in the bath, right? So you don't even try and remove it. So you saturate the system with norepinephrine. Even in that case, just deleting norepinephrine receptors from astrocytes or silencing astrocytes or doing other things that we found are essentially the astrocyte output downstream, all of these blocks the effect of norepinephrine on synaptic function entirely. It doesn't diminish it, it doesn't impair it, it just gets rid of it entirely. And when we found out about this, it turns out that we had colleagues working in the zebrafish with essentially the same story. And I think that's actually the most exciting part about all of this. It's not just one, of the bad discovery that someone's made in brain slices in the mouse. It's something that people have discovered in the fly, in the zebrafish, in the mouse. And what's interesting about these two is that these three organisms share a common ancestor that's about 250 million years old. And I'm sure you guys know this, and this is about when neuromodulators emerged, right, in the bilateral lineage. And that tells you right away that astrocytes and neuromodulators from the beginning have been playing hand in hand to modulate synaptic networks. So I think we're looking here at the very fundamental function of astrocytes and maybe a very fundamental interplay between neuromodulators and astrocytes, which is that these two things work together to modulate synaptic networks. So astrocytes are a part, in my point of view, of how neuromodulators achieve some of their effects on neural networks. I'm not saying all of them, evidently not, but at least the effect that they can have on synapses. And if you come at it from an astrocyte point of view, if you're someone who studied astrocytes for quite a while, it's actually so satisfying because we've known for 3 decades now that astrocytes have this close proximity with synapses. We know they contact almost all synapses, and we know they can do so many things at synapses. So it's very satisfying to finally find a way to show that this actually matters quite a lot. I mean, I can show you some slides if you want, but if you read the paper, I don't want to bother you with slides and images, but I can if you want.
I'm happy to see more. It's fascinating.
Oh, good.
I'm also curious, and maybe this didn't fall into the sphere of what you were looking at, but maybe we could talk about that first, and then we can go to the slides. So I've long been interested in the fact that astrocytes sit with their end feet on blood vessels, and the smooth muscle can control blood flow. And as a group that does a lot of fMRI work, we're constantly on the lookout for better understandings of actually what's going on in the connection between blood flow and neural activity. And there's this really fascinating kind of long-standing hypothesis. Chris Moore was the first to kind of really give it a name back in the day, in the mid sort of early 2000s. He called the hemoneural hypothesis. And the idea was that because neurons and blood vessels had essentially, been built together over evolutionary time, you couldn't really think of them as separate. If a neuron started to spike, then the blood flow that came in that allowed the neuron to then spike more was really deeply embedded within that whole circuit. Essentially, if you blocked blood flow, now the neuron couldn't do the things that it would do beforehand, and vice versa. The neurons had to trigger the blood flow to come in. So there was almost like an inextricably intertwined system. And so I wonder whether or not you have any thoughts in that space, or looked at all at whether or not the blood flow is important for the plasticity and might end up relating to some of the kind of neural hypotheses implications as well.
Yeah, this is not something I study, but I have several good friends who look at this. I have a kind of extreme view on this. I feel like I'm trying to always be the devil's advocate. You know, we've always thought of astrocytes as somewhat of a follower that does whatever neurons require. I think there's a equally probable way of looking at this, which is that astrocytes simply modulate how much energy neurons get, and therefore how much of the things they need to do they can actually do. There was a beautiful paper from my postdoc lab, not by me, but by another French person, actually. Jérôme is his name, Jérôme Classadont, where he showed that, for example, orexinergic neurons are completely under the control of lactate supply by astrocytes to keep firing at the rate at which they fire. And that astrocytes can essentially shut them down without killing them by just modulating how much lactate they provide. Because in fact, neurons don't really use glucose that much. They use primarily lactate, which is, well, lactate and pyruvate, which is given to them by astrocytes. And so you could have, of course, you could have the point of view that you have a constant flow, and if neurons need more, astrocytes open the valve more and you have more energy coming. But you can also have a more astrocyte-centric point of view, which is you might have a knob that says that you don't get to fire right now because I don't want you to. And the easiest way for me to do this is to not give you enough energy to fire at the rate you want to fire at. Right. And I think in that sense, the control of blood flow obviously, is intertwined with that. It's like you could modulate more extremely the amount of blood flow and therefore energy supply that comes in the system in the first place. But yeah, this is not something I study, but I think it's fascinating how we always forget about the astrocyte intermediate in fMRI readouts, right? We look at blood flow and we make conclusions about activity of neurons, when in fact, you might truly be looking at the activity of astrocytes there, that are the ones regulating the blood flow. And that doesn't have to be directly connected to what neurons are doing. I'm being anti-dogmatic here on purpose, right? But I like kind of challenge these dogmas. It's kind of my favorite thing to do.
I love it.
I know I'm not saying all of the fMRI work is wrong. I'm far
We
from me
just
to
need to
say this.
rethink about it. Yeah.
I think we need to rethink about it, because truly what you're looking at is blood flow, and blood flow is regulated by pericytes and astrocytes, not by neurons.
Yeah.
The activity of neurons might affect it, but what's actually regulating the diameter of the vessels here is astrocytes and pericytes, not neurons.
Not neurons themselves.
So how direct of a measurement of neuronal activity is fMRI? Question mark. And again,
Yes.
I don't
Well, I
like
think
to
we
work
all
in that
know
domain.
we all know it's indirect. We just like that it's also non-invasive.
Yeah, but I think we also like to ignore that it's indirect, right? We've all
Yes.
done this. We have the same sort of dogmas in our field. We look at calcium when we don't even know what calcium is actually doing in astrocytes. But I think it's important sometimes to keep in mind the fact that it is indirect. We should probably be more careful about that.
Yeah, so maybe we should spin a little further with this analogy, and I think maybe I can point towards where I think maybe even trying to think about either or is a mistake. So
Yes.
let's imagine now that the astrocytes are now controlling, as you say, whether or not the neurons get to be involved in the current process. We can maybe imagine via analogy, this is now a teacher in front of a classroom full of children. But now the astrocyte is the teacher. It is shushing the children. No, you can't speak right now. Okay, now it's a break time. You can have more discussions between yourselves, and now some information can be processed. You can imagine the context would matter, whether they have a visitor coming into the room with a message, could that propagate through the classroom? It'll depend on whether or not the teacher is shushing everyone or not. And you could then say, well, who's in control here? Is it the students in control of the message passing, or is it the teacher in control of the information flow? I would like to say that actually it's the classroom as a whole that matters, and that's what we should think about, is how is the classroom
Yeah.
functioning? And it's dependent on both. If you have a bunch of children that are full of food because they've just come in from recess and they're tired, they're not going to be wanting to pass many messages around. So it doesn't matter if the teacher's shushing. And if you've got children that have been sitting indoors because it's been raining for 6 straight days and they're full of energy, the teacher doesn't matter how much they shush them, the children are going to be loud and annoying. And so it's about the way that these things play together that we need to be thinking about, the emergent property of the circuit as a whole rather than one or the other. And I think what you're doing and I really admire is you're saying, hey, for a long time, all that anyone's cared about is that the children are talking or not. But guess what? There's all these other processes going on. There's even a principle. There's even other classrooms. There's even other scales that matter. And we need to think carefully about the context of all of this if we're going to come up with really sensible process models of how a brain actually works. it's going to take all these pieces to understand it in a fundamental level. Is that fair?
Yeah, that's a very good analogy. I like that. And I think you're totally right. It's never black and white, it's always gray. But I think it's the battle that makes us always land kind of in the middle, right? Every time there's a controversy, you always end up in the middle. And I think that's really where we're aiming at here. That's kind of why I like being the devil's advocate, because I know if I push that much, nobody's going to follow me, but hopefully they'll land somewhere in the middle.
Yeah, I'm the same. I used to play drums in high school, and my drum teacher said, as a drummer, you should always try to make mistakes as loud as you can, because at least then the whole band will have to follow you. And I've tried to do that.
Summer, how much do you reckon it's, I think this is like something you touch on in maybe in the perspective, but how much do you reckon we've sort of screwed ourselves with our knowledge about astrocytes by looking at them through this neuron lens where calcium is the, the big player that, everybody's sort of chasing and trying to make sense of it. Do you think that that's kind of led things astray? Do you think there's a lot less to uncover that will be, completely changed if we change the focus on calcium, which is such a kind of neuron bias?
Yeah, absolutely. I think you said it very, very elegantly that we went for calcium because in neurons, calcium is such a proxy of so many things. It's a proxy of depolarization. It's a proxy of an MD receptor opening at synapses and therefore plasticity. It's a proxy of, I have calcium, I'm going to have neurotransmitter release, right? So we went for calcium right away. We don't actually know what it does in astrocytes. It's a second messenger. So obviously, many things are going to be calcium dependent, but is that the signal we need to look at? Nobody knows. It's just the one we have. Thanks to all of you folks working on neurons, we have GCamp and things like this, right? So it's very convenient, and we found ways to make astrocytes express it. And it's told us a lot of things. It's told us, first of all, that astrocytes are active. They're not silent. They have a lot of calcium activity, which was a bit of a surprise from a neuroscience perspective, even though obviously every cell has calcium activity, right? So we had to wake up from that at some point, but it was still a surprise somehow, because in neuroscience, calcium used to mean excitability or activation or depolarization and things like this. But I'm really looking forward to the day that we look beyond calcium, because it's too neurocentric of an approach to look at astrocytes. So I'm guessing there's something more there than calcium to look at that's going to tell us a lot more about how astrocytes integrate information and then distribute it into defined locus or processes or something like this. I think there's definitely a lot more to do beyond calcium, for sure.
Can I ask a naive cellular question about astrocytes?
Yeah, of course.
Do they contain endoplasmic reticular like other eukaryotic?
Oh, yeah.
So they are storing calcium in the same way.
Yeah. This was for a long time the primary hypothesis for how calcium elevations or calcium signals are generated in astrocytes. So that led to this massive controversy in 2010, which is a beautiful pair of papers to do as Journal Club, honestly. You have one paper saying blocking calcium in astrocytes prevents synaptic plasticity. This is a seminal paper in Nature. And 2 months later, you have a paper in Science saying calcium in astrocytes does nothing. And this was because essentially the main mechanism that we thought was at play is actually fairly marginal in generating calcium activity in astrocytes, and that's the endoplasmic reticulum and IP3-dependent calcium signals. It does play a role, but it's not as important as we thought it was. So the way calcium signals occur in astrocytes is through a lot of things, apparently. Transmembrane calcium fluxes. You also have mitochondrial transition pore calcium activity, and you have ER, IP3-dependent calcium events, and then probably other things like plasma membrane pumps and stuff like that. It's very diverse. And we don't know if there's a rule about what sort of calcium signals happen where. Like, do you have just one type of channel mediating one type of event in some processes, and then ER-dependent calcium signals happening just in the soma or just in the branches? We have general ideas, but we don't know if there's a clear sort of landscape about the mechanisms that drive these signals.
And do we know whether or not in astrocytes you see the same massive difference in the concentration within a cell versus in the extracellular fluid? My understanding is that it's something like picomolar inside a cell and then millimolar outside in a typical eukaryotic cell. Is that the same for an astrocyte?
No, it's slightly higher. So astrocytes' basal calcium level is about 100 nanomolar. So it's about twice as high as neurons.
So it's
And that
not
depends on
really
what you're looking
as
at.
poised to notice a difference in calcium as
I mean,
a
that's still like what,
still a lot,
6 orders of magnitude difference, right? Something like that. I think neurons, the basal level of calcium in neurons is something like 15 nanomolar, I think,
okay.
isn't it? And it's about twice as much in astrocytes.
Okay.
So it's fairly comparable.
It's still pretty high. You would notice if calcium was released. Yeah.
Oh yeah, you notice it for sure. Yeah, probably have a couple of videos I could show you actually. Yeah, it's pretty obvious.
Sweet. Oh, if you've got videos handy, I love videos, but
Oh yeah.
yeah.
I just have to find them. Here we go. Let me share this. All right, so this is the classical scenario. We inject, you guys can see my screen, right?
Yep.
Yeah. We inject GCAM6 in the hippocampus with an astrocyte-specific promoter. So all astrocytes express GCAM, and then we do 2-photon microscopy. And so the movie I'm going to show you here is in the hippocampus. I outlined very roughly, literally on the PowerPoint, 3 astrocytes boundaries, very roughly. And then you're going to see a bunch of spontaneous calcium events, which are these colorful little flashes. And in this particular example, we apply norepinephrine. And so I think you'll be able to see the effect quite clearly. Let me play it again. So you see a lot of little flickering, which is a spontaneous activity, and then this big wave of activity is when norepinephrine hits the slice.
And so
And
you've
on
colored
this one,
the calcium waves as a different color depending on the different astrocyte that it's in?
This was done with the Aqua pipeline from the cancer lab. So it's showing you in individual events defined by the location and the time at which they occur.
Got it.
So
And
the pipeline
and then
allows
that's
you
where the
to,
color comes from. Okay, okay.
yes,
Can we see it
yeah.
again then, please?
Then I
Whoa.
have this movie here that doesn't have the norepinephrine wave, so you can see the spontaneous activity a lot more for a much longer period of time. And this is obviously
On the
sped up
right.
because,
Oh, sorry, go ahead.
no, go ahead.
I was going to ask, on the right is spontaneous activity. That's just what happens in terms of calcium transients kind of popping in and out. And on the left,
Yep.
if you evoked it with norepinephrine.
Yep, exactly.
Whoa. That's
So
really
there's really quite a lot happening, a lot more than we ever expected. And these movies are just showing you the obvious events, but most of them are actually very small. They happen on the scale of something like a micrometer square. And we can't really look beyond that because of diffraction limit of, light microscopy. But we suspect that these events actually happen on the scale of those tiny processes that contact synapses and other cellular elements. So really on the scale of a few hundreds of nanometers at best. And that's what people using STEM microscopy have found. So you can't have that information here using 2P, but we think that the smaller events really reflect those highly local calcium hotspots.
Amma, do you see in your data, I can't, if you would slow it down, do you see the that was shown kind of recently, this centripetal propagation where the calcium kind of goes, I got
Yeah,
that was quite controversial. So I wondered if you see it. And I guess
you know what? I don't think it's controversial. I think the data was very convincing. What's embarrassing is that, again, it's a very kind of neurocentric sort of idea that you have these events slowly making their way toward the soma, and then you have an integration of the soma. The thing that I find interesting in the paper is that they actually didn't show there was any integration. There is propagation of the events at the soma, but there is no evidence there is any sort of summation or integration there. And if there was, you would expect that maybe there would be then some centrifugal activity, and they don't see that. So my guess is that it probably has something to do with maybe gene expression regulation or things like this.
yeah,
We honestly have not looked at it in our data. We didn't look at all whether any of this activity propagates towards the soma over long time scales.
Yeah, cool. I listened in on a talk, I don't know, maybe 2 weeks ago with the first author of that paper, and somebody asked the question, is it just because in those distal processes, there's sort of more or less of the GCAMP, and so you just naturally see a kind of a different sort of pattern of emergence of activity. So I guess that's something that comes down to it too.
Yeah, the time scale at which they see the propagation is I think something like 5 seconds.
Yeah.
Which seems really
5 seconds. It takes 5 seconds to go from
It takes about 5 seconds, yeah.
Wow.
Yeah.
Slow.
From the tip of the processes to the soma, I think, if I remember well from the paper. So it's really quite slow. I don't think that has to do with some sort of artifactual GCamp expression or anything like this. Yeah. Yeah.
So I guess we could open it up for some questions now from the students. There are a couple of lines of inquiry. Julia, did you have your question answered through the process, or do you want to come back to that?
Yeah, thank you, Mike.
Questions.
It was a wonderful, not talk, I don't know what to call it, conversation. I have lots of questions, and now I forgot most of them. So I'll start from recency effect from the last one I had as you were talking now. Coming from an fMRI background, so hopping on what Matt was saying about the neurovascular unit not being, an fMRI, we just primarily look at what we think it's neuronal, but we don't know what's going on. One question that I had was, given all the information you gave us now in astrocytes, how would you image then this neurovascular unit? And the reason why I'm asking this is because from what I gathered, there's no such thing as a common denominator of all members that are involved in the neurovascular unit. So now you're saying that, astrocytes and neurons evolve over different timescales. There's different mechanisms that may be at bay in the different members. So what is the ground truth and how do we even get there? If everyone is playing their own game. And so it's that's what I was trying to get at. So that's one question.
That's a good one. I don't have an answer to that. I feel like you have to figure it out. That's your job.
Oh, thank you.
No, it's a very good question. I really wish I had a good answer to that. I think at the very least, as long as you keep in mind what are the actual cellular mechanisms at play underlying what you're looking at, I think at the very least, that's going to make you a lot more careful in your interpretations and how you discuss your data, which is something that maybe more people should do. In terms of actually imaging all of the different players in humans, because I'm assuming you're doing this in humans, right? Yeah, I don't know. Man, I wish there was a way to look separately at neuronal activity, astrocytes activity, and then the actual fMRI signal all together in a human. Goodness, we wouldn't have to use mice anymore.
Mm-hmm. Yeah, that's a good point. And then I have another question. Because one thing that was, kind of like I had a wow moment, and it's when you said that there's a one-to-many relationship between astrocytes and neurons. So my mind immediately went to think how disastrous it is when an astrocyte is not functioning properly. If it's such a thing that there is no resilience in this astrocytic network, if we want to call it that way. Because if one astrocyte is hit, then as you're saying, is the neuron getting any of its nutrients? If it was provided by that astrocyte and now it's not working properly. And so that made me think, if it's a chicken and an egg kind of problem here, what came first, and maybe the neuron could still, provide whatever function it had, because it learned something from the astrocyte and it can provide for himself. I don't know what your take on that is.
Yeah, that's another very good question. I think if you just kill off one astrocyte, you might not really see an effect at the scale of an entire network, because just the few chunks of dendrites or a few hundreds of thousands of synapses that the astrocyte contacts, or just that within the entire network might not contribute that much. I guess for some functions, we're talking about emergent properties. So it's the summation of many neural elements doing the same thing that makes the function emerge. So if you lose a tiny bit of it, I feel like there is a sort of resilience that built in. But now, if you're looking at just the outcome of killing off the one astrocyte from just a cellular point of view to these neuronal elements, yeah, the consequences might be quite dire. We know that, for example, synaptic plasticity requires signals coming from astrocytes. So presumably, if you don't have the astrocyte anymore, you wouldn't be able to do LTP or LTD or things like this at the synapses. You wouldn't necessarily be able to feed the neurons as well. Yeah, that's a very good point. And honestly, I don't think it's going to be a surprise when we start seeing a lot more data suggesting that maybe many neuroscience disorders actually come from glia, which is a prediction that was made 200 years ago by most of the neuroscience pioneers. Rudolf Virchow, Ramon Cajal, all of these guys, they actually predicted that many of the diseases of the brain were diseases of the glia. They were just making this assumption based on observations that glial cells, primarily microglia and astrocytes, were dramatically changed morphologically in samples from disease patients, much more than neurons. So morphology doesn't tell you everything, but they made a very interesting hypothesis there that maybe the diseases of the brain were diseases of the glia. And I think that I don't know, I feel like it's an interesting idea, and I wouldn't be surprised if we see a lot of data going this way. You could also look at it the other way around, which is we focused on neurons exclusively for the past 70 years. How many brain disorders do we cure nowadays?
So,
Right?
yeah, I mean, not many. Thomas, that actually, the morphology change actually dovetails with a question that Annie had put into our Slack earlier, and I wonder if Annie, you wanted to pick up that thread and continue with it, because it's absolutely fascinating to think about morphological changes. Annie, do you want to?
Yeah, same wavelength. Thanks, Mac, and thanks, Thomas, for a fantastic talk. Yeah, my background and limited exposure to glia is from the perspective of neurodegeneration and inflammation. And in histology, as you would well know, we see that astrocytes and microglia, after protracted inflammation, start to kind of contract their processes and develop this like more reactive morphology. And I was really interested in the point you made about how we see this sort of decentralization, where in the tripartite synapse, it's these very localized interactions, but there's some sort of centralization happening, or else we would have seen the cells divvy off into many independent cells rather than staying connected. I'm wondering if you have a sense of the reason that we see in protracted neuroinflammation, this sort of drawing in of processes is an attempt to get a more like all hands on deck centralized response to pathology. Is that something that you think about?
Yeah, that's a good question too. So we don't study this sort of, we don't study neurodegeneration, so I don't really have my hands on the pulse here. But I think a question related to what you're asking is whether the phenotype you see in glial cells during such disorders is an attempt to compensate for what's happening, or a way through which the disease is really occurring and causing all of the pathology of the neural networks at large. So is this a reaction of astrocytes, or is it the cause of the disease? And quite frankly, I don't think anyone knows, because we don't currently have ways to cause this sort of phenotype in astrocytes and see downstream, is that causing a disease or not? So when we study these sort of neurodegenerative disorders and look at astrocyte reactivity, it's a good marker that something is happening. But quite frankly, nobody at this time that I know of would be able to tell you if this is beneficial or pathological in itself. Does that make sense?
Yeah, perhaps an inverted U shape, like we think
Yeah.
about a lot in the lab, of initial benefit and then
Mm-hmm.
eventually comes full circle to become deleterious. But also a chicken or the egg
Yeah.
situation, like Julia was talking about.
I think there was all the work from Shane Littlelow, who was at NYU, with these, A1, A2 astrocyte types, that suggested that astrocytes that are reactive are no longer protective and become deleterious to neurons. But I think this has been largely revisited to some extent, right, by the literature, by people in the field. So I think the question is still up for debate. Like, are astrocytes trying to help the system, or are they causing the disease eventually when they become reactive like this? I really don't know. I guess that's another question for you guys.
For what it's worth, Toma and Annie, there's a beautiful developing interest in neurodegenerative disorders and neuroinflammatory disorders in actually kind of trying to treat the diseases and actually find disease modifying effects using neurogenetic drugs, with the idea being, that they're kind of that their effects on disease modification are going to be via microglia and astrocytes. And it seems to be getting a lot of attention in the field, and there's a lot of work in rodents and even some emerging stuff happening in humans. So I think the field is paying attention to it from that angle. So
on the topic of triggering astrocytes with noradrenaline, Jason, you had some questions that related to that, if you wanted to unmute and ask.
Yeah, sure. Yeah, so I was wondering if you could say a bit more about how the astrocytes get inputs and how specific those inputs can get. I kind of get the sense that the astrocytes get bathed by noradrenaline from blood vessels or from the general extracellular milieu, but can they get can inputs talk to a specific astrocyte and not other ones? Can input talk to just one part of a specific astrocyte? How specific does it get? And my interest in this question is because I want to figure out my questions are around computation, basically, and how astrocytes might
Oh, a really cool area there. Yeah. I guess that links back to I think a conversation we had at some point that nobody has done the key experiment of photo-uncaging or half applying norepinephrine just very locally on the astrocyte and see how the astrocyte reacts. So the short answer is we don't know. We haven't done that experiment in the lab, and I'm dying to do it. Just uncage norepinephrine and see, do I get one tiny hotspot of calcium activity there just where I uncaged it, or do I trigger the entire astrocyte and then the entire network? I don't know. I really don't. And then the question also gets to what do you use to stimulate the astrocyte? You might see something with norepinephrine, and you might see something completely different with another neuromodulators or even a neurotransmitter. So then you get to the question of what sort of receptors do astrocytes have? And by extension, what type of astrocyte am I looking at? Am I looking at an astrocyte in the hippocampus, an astrocyte in the spinal cord? Because they might have different receptors and therefore react differently to different stimuli. So one thing that we're crucially lacking right now is essentially a map of inputs that astrocytes can respond to or that they can sense and how they respond to them. And that would be a very cool thing to do, actually, get a full input-output sort of map, functional map of what astrocytes react to and how and when and where. We don't have that, so sorry.
That would be very cool. I mean, I'm working on the microns data set, and this seems like the kind of question one can ask of the microns data set, like just looking at, what synapses and cells an astrocyte's talking to.
Yeah, I don't think they have the resolution though, because if I remember well, their actual thickness was 40 or 60 nanometers, right? And you lose most of these astrocyte synapse interactions if you go beyond, I think it's 30 or 40 nanometers. So if you really want to capture all of the fine anatomy of astrocytes and how they interact with synapses, you need to actually do something much more refined than what they did, believe it or not.
Yeah.
So you might not have all of the data that you need to really get a map of how astrocytes contact synapses and what synapses and with what logic. And also you're going to miss all of the molecular stuff there because they didn't do any like spatial transcriptomic on this data set. So you don't know what receptors are expressed or where or how many of them. In fact, they didn't do calcium recordings either. So you really only have the backbone or the structure of the astrocyte there in the micron data set without any sort of functional or molecular annotations to it, which is a big disappointment. But yeah, I think
That's how you know you're an aficionado. You take a look at the micron data set and you go, nah, it needs to go even more fine.
I wrote to them 3 years ago because their merge split errors for astrocyte segmentation is just horrendous. Half of the astrocytes are misclassified as blood vessels. And it's just so obvious. You just click on a blood vessel and you see this magnificent, anastomosis of blood vessels. And then you see these weird, like little bushy things. You're like, oh gosh, these are just all astrocytes. And I mean, granted, it's extremely hard to correct merge split errors when it comes to astrocytes, especially when nobody in the micron consortium has any expertise with glial cells, but all of them are neuron people.
Yes.
So yeah, there is so much more to do there. I'm very frustrated by this project.
I've seen a few cells that are astrocytes mixed with neurons, and they look interestingly crazy and easy to tell apart.
The first time I looked at the data set, I was like, oh my goodness, they have astrocytes with some sort of axons coming out of them. This is so cool. And then quickly it became not cool at all because I realized this was just merge split errors. But this was actually exciting because in the humans, astrocytes do have this sort of long processes that span the entire cortex. You have what we call the velate astrocytes in layer 1 of the cortex that send projections all the way through the bottom of the cortex. Nobody knows what they do.
Velate?
I can't really. Yeah, some of them are called velate astrocytes, and then you have another class called varicosity astrocytes that are, I think, in layer 5 and 6, and they send something that looks exactly like what you would expect to be a norepinephrine fiber, with varicosities along the way, like these big bulges, which makes you wonder, like, are these like fibers or axon-like structures from astrocytes that can release stuff? And obviously, nobody knows. All we know is how they look like. But so when I first looked at the data set from the Macrons consortium, I was like, oh my goodness, the mice have it too. How come we've missed it? Turns out this is just axons that are misclassified as an astrocyte process.
And forgive my ignorance, but how do we know that these fancy astrocytes in humans are actually astrocytes and not a misclassified neuron?
Yeah, we don't know actually. So this data is almost exclusively GFAP staining from human tissue and primate tissue. So I think it's reasonable to think that this is astrocytes, an astrocyte element, because it has a GFAP stain in it, but you're right that it might actually not be at all. The only data we have is, like, I think there's maybe a dozen of papers that looked at brains from a variety of primates, and then you have a lot of data from human tissue that sees these sort of projections from astrocytes. This is very consistent, and then there's quantifications about what type of monkeys have them and what type of primates have them, and etc. So this is seen very consistently, but you're totally right that it might actually not be a part of the astrocyte. It could be a sort of projection from, I don't know, the locus coeruleus that somehow contacts the astrocyte, and that alone is sufficient to show up on the
Pass
IHC.
information or something. Yeah. I learned about this recently. I have an interest in lots of different parts of the brain, but the cerebellar nuclei, we're in a very different location now, but people don't really know exactly where the cells there project, whether or not they're targeted or spread. There's all this complexity there, and it's a very hard region to study. And one of the only studies we have with using modern techniques used an AAV, so like an attenuated virus, injected them into the cells there, and then looked at and see later, a couple of days later, where the different viruses had spread to. And it turned out that, depending on where you injected, let's say there's like 100 cells that they injected, the patterns that they saw downstream were actually really, really similar. In fact, they looked almost identical to one another. And so you might be wanting to conclude that essentially all the neurons project to exactly the same locations. But if you go and look at that type of AAV they use, it was an AAV8, they've actually got a lot of transcytotic spread. So that means that if you inject it into one cell, it's going to spread into other cells, and particularly if those cells have got gap junctions, which cerebellar nuclei also have. So you've got this really huge challenge now where you need this incredible rich technology to get at this amazing access, but it has these caveats to it and these confidence intervals around it that you really have to be careful about the conclusions you make from it. And I think we have to remember this, particularly as tourists in these fields that don't use these techniques on a day-to-day basis, that it's really, really hard to make these conclusions and be confident about them. You have to have a grain of salt with everything you do in this field. But it's exciting times.
Yeah, I agree. You always have to be wary about the technology you use and whether it's actually as specific or efficient as you think it is. And I think that's true for so many things.
Oh, yeah.
Like even up to genetics, right? People have figured out that you have opsins naturally expressed in some elements of the brain, and when you send the light, even without expressing general opsin, you might have an effect, right? Things like this.
I didn't know that.
I think, yeah, it's work from Serge Charpak in Paris, that you have effects on brain tissue from wild-type mouse by just shining blue light.
Oh my
And then
God.
you have other work showing that if you shine too much blue light, that alone hits the tissue and therefore depolarizes neurons, and therefore you have an effect on the network without any opsins in the system. So I feel like every time we introduce a new technique, it's like, oh, this is the best new thing. And then it takes 5 or 10 years to realize, oh, actually, it's got a lot of caveats too, right?
It also makes me wonder what blind watchmaker would ever put light sensitive cells on deep inside of a brain. But
I think it was the blood vessels, actually.
okay, there you go. Do we have other questions from the audience? Anyone that has a burning question for Tamar? I have a fun prediction for you, which I'm not sure if you're interested in testing, but I just love looking at these canopy-like cells in the astrocytes in the cortex, and you can't help but wonder that if you removed one astrocyte from that canopy, the way that in a forest now, because there's light to be had, the trees sort of fill in the gaps and then basically take over that space. I wonder if you test that in in a slice. You could go and remove a single astrocyte if you could do that delicately, and then watch over time to see whether or not the gap fills in, and whether or not there's processes at play. Because then you can imagine maybe there's particular molecular markers at play that say, hey, we need help. There's no astrocytes helping us. This classroom has lots of children and no teacher. Quickly bring a substitute teacher in, and, or make the teacher spread out over the class or something. I just think that would be a really elegant way to solve this problem for a brain, essentially to have this need for astrocytes, but not so much worry exactly which one you had. And I wonder if you think
Yeah.
that's testable.
Yeah, I don't think you could do it mechanically, like you couldn't remove the astrocyte, but you could definitely engineer a system where some astrocytes express, for example, diphtheria toxin. So you could kill them on demand in a genetic way, and then you could just have sparse expression of the Cree or sparse low tamoxifen injection, and basically have sparse activation of the construct, and some astrocytes here and there would die, and then you would see a ghost for a while anyway, presumably of the astrocyte not being there anymore, and then you could see if the ones nearby would take over the territory.
Well,
I agree. It's
if you ever
likely
test that, let me know what the answer is. I'd love to know.
Yeah, no, I think it's a very cool thing to do. Actually, I'm surprised nobody has done it because it's also technically not that challenging. All the tools are there.
Oh, sweet, awesome.
Yeah.
Brandon, did you have a question? You just videoed.
Yeah. Thanks for the talk. I was just kind of curious with like some the so you're talking about like the reconfiguring circuits of astrocytes. Do you know if like astrocytes, like one astrocyte will sort of, cover cells with similar receptive fields, or do they sort of cross across cells with different receptive fields? Like, is it sort of like, is there a function there?
Yeah, that's a very good question too. This is where we really need a better talk between hardcore neuroscience people and glial people, because I don't think it's something that anyone in my field has ever done. I can't think of a single paper that looked at that. This is such a good question. Let me think for a second. No, I can't think of any paper that looked at that. But yeah, definitely that's something that needs to be done. Absolutely.
Yeah. Oh, cool. Thanks. And then
Especially if you look back to the idea that Maxwell was saying about, feed-forward inhibition and maybe shaping the receptive fields and things like this. Yeah, absolutely.
Yeah, I mean, it seems like it'd be a really interesting question. Like, I'm just curious if that's followed up. And then the other quick question.
You can extend this to so many different things, like even the idea of place cells.
Yeah.
Place cells is something that is so context dependent by essence. How much of the grid of place cells is shaped by astrocyte modulation, direct or indirect, right? Sorry, I cut you off.
No. Yeah, I agree. Like, I mean, place cells would be perfect for it, right? Like, that's a great question. The other one was just with the sort of your contextual guidance framework that you're putting, like your argument. I wasn't sure when I was skimming the paper. It's sort of, you have in your contextual guidance, the modulatory input goes into the astrocytes, and like, is it sort of like assuming the previous version where it also goes to the neurons, or because like in the caption it sort of says they're not synaptic by default, but I wasn't sure if I misinterpreted that as saying. You're saying the main effect is through the astrocytes. Is that correct?
So, while this was, again, us being the devil's advocate here, we made the hypothesis that many of the modulatory effects that eventually affect networks have to signal through astrocytes. Obviously, not all of them will. When you apply norepinephrine on a slice, or when you release norepinephrine in the brain tissue, obviously not all of it goes to the astrocyte, because we know neurons have receptors, right, for norepinephrine, obviously. So some effects are going to be direct. That's pretty obvious. But in terms of modulating synapses, maybe they're not. And so that's what the preprint we have right now will be published next week, actually. That's really what we show, and I think you can easily extend that to other neuromodulators, like dopamine, for instance. We know dopamine affects synapses. It actually inhibits them very often, just like norepinephrine. And like always in synaptic physiology, if you have something that has an effect on synapses and then you abolish the effect with the receptor antagonist, your conclusion is the receptor is there on the synapse. That's how we've done neuroscience for the past 70 years. What we found, and I think that's the strongest point about this paper, suggests that we need to rethink that. Because you could easily have an astrocyte intermediate there, sensing dopamine, sensing histamine, sensing serotonin, whatever you want, and actually enacting the effect on synapses, and then they can become synapse specific as well, right? But obviously, you're right that the effect of neuromodulators overall is going to also be direct to some extent. But as for our synapses, my bet would be that most of it is still astrocytes.
Okay, great. Awesome. I'll read the preprint. I think that's not the answer.
All right. If there's no other burning questions, we might start to wrap up. I wanted to give, before we thank you and send you on your way, Thomas, because it was so awesome and so much fun. Is there anything that you think that, we didn't cover, like one last little nugget of speculation that you think would be, you think it's super cool to mention, or, any wild, your chance for sort of wild speculation or something that you think that we should know about that hasn't come up?
No, I think you guys touched on everything, actually. Physiology, morphology, neuromodulators, a bit of hormones and lactate, and the microns consortium data set. You covered a lot.
Great. And I
Even
mean,
fMRI. No, that was great. That was a lot of very good questions.
I mean, I, for one, I don't, neurons are dead to me now. I don't care at all about them. So I'm sold. I hope everybody else is.
I'm all about the interstitial space myself.
Oh, okay.
I think that I like that you mentioned that. I think this is something that we overlook largely, even in the way we present it in schematics and textbooks. The interstitial space as being this loose, just sort of like large volume, completely unregulated. That's completely bonkers. First of all, there is not that much space between the cells. And second of all, it's highly compartmentalized. I think it's not one homogeneous space, it's just a bunch of small units. And these units are going to be very important because this is where the communication happens between different cells. Right? They don't touch each other on the shoulder, they talk through the interstitial space. So that space has to be highly regulated and highly controlled in terms of its sphere of influence, where it is, how large or small it is, because that alone is going to change concentration, therefore activation of receptors and things like this. So there was this very cool paper in Nature, I think a couple of years ago, from C. elegans, where they did super complicated work, but the conclusion was that half of the communication that happened was actually not synaptic. It wasn't wired communication, it was non-synaptic, extrasynaptic communication. They didn't quite put a name on it, but I think you could rephrase this by saying this is glial communication or interstitial communication. So yeah, the very neurocentric, synapse-centric view of brain function, like the one that is essentially outlined in the Microns consortium, I think is dangerous to still follow nowadays in 2025.
Yeah, awesome. I think that's a perfect message to end on. Thank you so much. This was great. You were really generous with your time, and we learned heaps. Hopefully it, felt nice and free flowing, and yeah, it was great for us.
That was my pleasure. So if you have any questions, if I missed anything, please let me know.
Yeah, for sure.
And if you need to edit the video for, if you need to slow it down, that's fine.
Probably all of us. Yeah, we all need to be slowed down, I think. But yeah, thanks again. This was so exciting and such a fun conversation.
Yeah, my pleasure. Thanks for the invite.
Bye.
Bye-bye.
Bye.